Yes, madam. You can continue.
Yeah. So my presentation is divided into two parts. The first half an hour, I'll be explaining the topics, that is overview of drug repurposing, why we do the drug repurposing, and different approaches in drug repurposing. That is, there are two approaches: computational approaches and experimental approaches. Why we go for drug repurposing, what are the main advantages, and yes, of course, drug repurposing has some challenges. So with this, the first part will be over, and the second part will be explained. That is one case study. Actually, one of my scholar, Mr. Nagaraj, he carried out extensively in silico drug repurposing using different online and offline tools. So with that, my presentation will be over. Yeah. So what is drug repurposing? Actually, according to James Black, the most fruitful basis of the discovery of a new drug is to start with an old drug. So drug repurposing represents an attractive drug discovery strategy. So here what we do, actually this drug discovery, it starts with polypharmacology, and we know very well traditional drug discovery is, or conventional drug discovery or drug design, always we focus on one drug, one target. Okay, so if I am working for discovering an anti-cancer drug, I have to find out the proper pathway, and I have to find out a target, and I do some sort of designing followed by laboratory work. But this drug repurposing, completely it will be on multi-target.
Madam.
Hello.
Yes, madam. It is audible.
Yeah, Swati ma'am, like all the things are going correct?
Yes, madam.
Yeah.
All the things are
So
going.
as we all know, so the drug discovery, it starts with the initial phase of discovery and development, then preclinical research, okay, followed by clinical research, FDA review, and FDA post-market safety monitoring. So when we come to this drug repurposing, maybe this 3 to 4 years of the drug discovery process will be less, because here we are taking the drug which is already tested for its safety and efficacy. Okay, so only the thing is the use is intended for some other purpose. Okay, so here the discovery phases, it will start like this: 1 to 2 years compound identification from approved drug library, and the next will be we have to validate the drug target and preclinical research. After, followed by that, the first 1 to 6 years it will be the clinical studies, and followed by registration. So here what I wanted to tell is, we are playing with the de-risked compounds, because we are not taking the we are not doing the discovery from initial, we are not doing the discovery from the scratch. Okay, so we know very well the drug discovery, though it is very complex process, though the CAD tools, everything comes, again we are suffering with a high attrition, and the drug discovery is a slow pace, and lot of money is involved. So this drug repurposing, it can replace, or it can reduce the years and cost, as well as the manpower also, it will be reduced. Okay. Yeah, and coming to the advantages, this is very, very important. So the risk of failure is lower, because the repurposed drug has already been found to be sufficiently safe in preclinical models and humans. So we can skip the phase 1 trials have been completed, because we assuming that the compound, the drug is already proven for its safety. Okay. The second one is, as I told you, 3 to 4 years of time frame for drug development can be reduced, because most of the preclinical testing, safety assessment, and in some cases substantial savings in preclinicals and phase 1 and phase 2 cost. Okay, as I told you, indeed the costs of bringing a repurposed drug to market have been estimated to be around $300 million on average, compared with an estimated 2 to 3 billion for a new chemical entity. And very important advantages, the repurposed drugs may reveal new targets and pathways that can be further exploited. That's what I told you. It is always depending on polypharmacology and multiple targets rather than a single target. Where the drug repurposing started, okay, the origin. Okay, so the drug repurposing is based on polypharmacology, a field of study related to design, discovery, synthesis and use of pharmaceutical agents like small molecules, drugs, antibodies, stabilized peptides, natural products that act on multiple targets. Again and again I am stressing, so these two words are very important in the field of drug repurposing, where you will be playing with so many targets, okay, and you will be playing with multiple pathways. Okay, and we know very well why this multiple targets, because the complex diseases such as neurodegenerative disease like Alzheimer's disease and some like anti-inflammatory disease, rheumatoid arthritis, so and cancers, so they all involve multiple genes, the networks of multiple genes, hence necessitate multi-pharmacophores as part of their therapeutic and system-based approaches. So one such example is multiple pathways that is which is involved in the rheumatoid arthritis. You can see here, if you see the rheumatoid arthritis, usually the pro-inflammatory cytokines, they will be released during the inflammatory conditions like IL-2, IL-6, TNF alpha, like that. Okay, so each and every cytokines, it what I can say, it regulate a particular pathway. Okay, so for example, if I go for this IL-6 during the inflammation, IL-6 will be high, so it activates the JAK-STAT pathway. Correct. So similar way, one other pathway is P13K signaling pathway, then MAPK signaling pathway. So what I wanted to mean here is a disease which involved in multiple targets, and there will be complex pathway. The pathways are very complex in particular disease. One such example is rheumatoid arthritis. It may go for Alzheimer's disease. If you take cancer, any disease, it will go. Now, the second very important thing is off-target effects paved the way for repurposing. Okay, so again the repurposing, it all started with accidental or serendipity discovery. Okay, so as I told you, the pharmaceutical agents can interact with multiple targets. So unintended interactions between drug and off targets could result in toxicities or side effects that we all know very well. So in that, any one of two off-target effect, it may lead to a significant biological activity or significant pharmacological activity. So very good example is sildenafil. Okay, so sildenafil is well known for it is reducing the hypertension. Actually, originally it is intended for the treatment of hypertension. Okay, so one of its off-target effect is it is used for the treatment of male erectile dysfunction. So we know very well it is known as Viagra, which is what initially found by the Pfizer's company. So here the original intended is anti-hypertensive, but the off-target effect of the sildenafil, it is lead to the male erectile dysfunction. So the off-target effects, it paved the way for drug. Another one example is, we know very well, thalidomide. Okay, so thalidomide actually originally it is intended for, it is a very good sedative. Okay, but unfortunately the drug, it has been withdrawn because the pregnant women, during the first trimesters, they have taken this drug. Unfortunately, the children, they were born with severe skeletal birth defects. So the drug has been withdrawn. But notably, it was withdrawn in 1950s, but in 1960s, it was used for the treatment of erythema nodosum leprosum. Okay, so it is a condition, it is a multi-organ failure, okay, due to the severe condition of leprosy. So the drug is used for the treatment of that. Later on in 1999, it is used for the treatment of myeloma. Okay, then the another one, the derivative of this thalidomide, that is lenalidomide. So again, this lenalidomide is also used for the treatment of multiple myeloma. So here again I am stressing the not the on target effect is the sedative effect. Other than on target effect, the off target effects paved the way for this treatment of erythema nodosum leprosum or later on multiple myeloma. So these are some successful stories of drug repurposing. It started in the year of 1987 itself. The zidovudine, actually the original indication, it was intended for cancer, actually anti-metabolite. Now it is used for HIV and AIDS. Then minoxidil, we know very well, it is hypertension, anti-hypertensive drug, but it is used for the treatment of hair loss. Then sildenafil, just now we have seen, and thalidomide, it was used for morning sickness and sedative, but now it is used for ENL and multiple myeloma. Celecoxib also, it is a very good COX-2 inhibitor. Now it is used for familial adenomatous polyps, like atomaxetine, duloxetine, like that. So many drugs, rituximab, relaxifan is very well repurposed for the treatment of breast cancer. Then topiramate is used for epilepsy, now it is used for obesity. Then ketoconazole, actually it is a well-known azole antifungal drug, now it is used for Cushing syndrome. Like that, we have so many successful stories of drug repurposing. Now, very importantly, what are all the approaches, okay, how we take further the drug repurposing. So the majorly this drug repurposing is divided into 3 phases. The first phase is very important phase, identification of a candidate molecule for a given indication. Okay, so here we have to generate the hypothesis, because hypothesis generation is very important. We have to understand the mechanisms on the various pathways involved, associated genes involved, or associated proteins, enzymes, what and all involved in the around the particular disease. Okay, so the modern approaches. So here the various computational tools, the molecular docking, networking. Okay, so all these transcriptomic signatures matching, all these computational tools are involved in the first step. Okay, so the hypothesis generation is very crucial in case of the drug repurposing. And the second one is, of course, we need to do the mechanistic assessment, okay, in preclinical models and some in vitro models, and of course it has to be validated with preclinical models. And the step 3 is evaluation of efficacy in phase 2 clinical trials. Okay, as I told earlier, assuming that there is sufficient safety data from phase 1 studies undertaken as part of the original indication. So this is the general approach for drug repurposing. Now, the drug repurposing is broadly divided into 2 categories. One is in silico repurposing, other one is activity based in silico repurposing. So here the in silico repurposing we group under computational approaches, and the activity based in silico repurposing we group under experimental approaches. Okay, so the in silico repurposing, what we do, we know very well from the protein data bank, we get the crystallographic protein structures. Okay, and the drugs, suppose the existing drugs, no, you can get from the drug bank. Then you do the molecular docking, the well-known structure-based drug design. Okay, so the molecular docking is a preliminary testing. Of course, you have to validate the molecular docking by doing the molecular dynamics. Then you have to do the MMGB assay and MMPB assay, that is the free energy binding assay. At last, you have to do the analysis. Okay, so on the right hand side, no, the right hand side I have the activity based in silico repurposing. So here like without computational approaches, directly I can take the drug and I will go with some in vitro and in vivo models. For example, here anti-cancer, first we are checking with the MTT, the cytotoxicity, then flow cytometry for cell cycle analysis, and finally the in vivo methods. Okay, so here as I told you, the computational methods, no, it utilizes the publicly available databases and various bioinformatic tools to systematically identify and we can go for the network analysis also. We can construct the protein-protein network and protein-drug network, okay, between drugs and protein targets. Yeah, so these are the different approaches. Okay, any one can be chosen according to your need. Okay, according to your aim, according to your hypothesis. Okay, so the down one, the yellow color is 2 types of experimental based drug repurposing. One is the binding assays. Okay, you suppose you found out the proper target or proper enzyme, directly you can go with the enzyme binding assay. And another one is phenotype screening. Here you go with high throughput phenotype screening, okay, using in vivo and in vitro model. Remaining the blue color is computational methods. There are these many computational methods which are very well available for in silico repurposing, okay, starting with genetic association. So definitely each and every disease, like there will be mutations involved, a particular gene, okay. So you can find out the genetic setup of that particular disease and you can match with the different targets. Then coming to the pathway mapping. So this is very interesting. You can construct the networks, okay, with you can first find out the various genes or various targets, then you can do the construct the networks, then you can finalize the targets. After finalizing the targets, that particular targets you can map with different drugs, okay. Then you can go for molecular docking. I think nothing much to explain that. It is a well-known structure-based computational strategy to predict the binding site complementarity. So always there will be two aim or two things that should be in your mind when you do the molecular docking. One is the binding site complementarity. That means the interactions, two-dimensional interactions compared to the co-crystal, and other one is the binding energy. Okay, so that you can predict the binding affinity. Then retrospective clinical analysis. Even you can, if you have the what I can say electronic health records, you can monitor the adverse effects and you can match with that. Okay, so that also will pave the way for the drug repurposing. And signature matching. Okay, so the signature matching, it may be the transcriptomic the association of the drug or disease, or the simple chemical structure, simple scaffold mapping or scaffold matching, or adverse effects matching. So these are all the signature matching. Okay, then the novel data sources, large scale in vitro drug screens with paired genomic data. So these are all the various approaches used in silico repurposing. Okay, starting with docking, genetic association, pathway mapping, retrospective clinical analysis, and signature matching. Now let us see one by one very quickly and briefly. So the first one is signature matching. Okay, so as I told you, what is signature matching? It is based on the comparison of the unique characteristics or signature of a drug against that of another drug or disease or clinical phenotype. Now, what are the unique characteristics? When we call that as a signature, okay, so that unique characteristic, it may be transcriptomic, proteomic, or metabolomic data. It may be a chemical structure. It may be adverse event profile. Okay, so these unique characters you can map between drug and drug or drug and disease. Okay, so that is called as signature matching. Yeah. So used to make drug disease comparisons, that is estimating drug disease similarity and drug comparisons. As I told you, drug similarity. You can see here, this is one typical example of gene expression. Okay, normal condition, and when the gene is overexpressed, when the gene expression is under expressed. Okay, so for this, you can use so many like NCBI omnibus one database is there, connectivity map one is there. So these are all will give you the publicly accessible gene expression databases. These are all. It will give you the complete details of the particular genes. Okay, you will get the transcriptomic signatures. Okay, so this is one such way. And another one is you can go for drug disease similarity and drug similarity. It comes under the same thing, that is transcriptomic signature. Okay, so what is drug disease similarity? The transcriptomic signature of a particular drug is derived by comparing the gene expression profile. That's what I have seen in the previous slide. You can compare the expression, gene expression of disease. Like before the drug has been given, what is the expression of the gene, and after the drug has been given, what is the expression levels. Okay, you can compare that. Okay, so that gene expression, it may be from a cell or a tissue, whatever it is. Then once you got the gene expression, the resultant differential gene expression signature is then compared with the disease associated expression profile that has been similarly obtained through differential expression analysis of disease versus healthy conditions. Okay, one such example is topiramate. Okay, so this topiramate is an anti-epileptic drug. Okay, so when they have compared the transcriptomic especially on gamma amino butyric acid. Now the drug is very well used in inflammatory bowel disorder. That is a very good example for the success story of this drug disease similarity. So as I told you, here they have used two databases. One is NCBI, gene expression omnibus. It gives the gene expression signatures of the disease. Then CMAP, that is connectivity map, it gives the gene expression profile of 160 drugs. So what they did, they have taken the inflammatory bowel disease genes associated with that. Then they mapped with these 160 drugs and found out topiramate is used for the treatment of this inflammatory bowel disease. Now coming to drug similarity. So again, this is aimed to identify shared mechanisms of action of otherwise action. What is the common understanding is drugs, it may belong to same class or it may belong to different class. It is not necessary that they should have the common structural similarity. Only thing is a common shared transcriptomic signatures between two drugs. That should be the similarity. So it can imply that it is common understanding that they also share a therapeutic application commonly. Okay, regardless of the similarity or dissimilarity in their structures. Okay, so fasudil, it is a Rho kinase inhibitor, ROCK inhibitor. Now it is used for the amyotrophic lateral sclerosis. Now, based on the chemical structures. Why not the chemical structures? So this we do very usually when we design our project for our UG and PG students. We used to compare the chemical signature. It's nothing but the structural features. SAR, we analyze the common different structural features. So comparing the chemical signature of one drug with that of another drug to see whether there are chemical similarities could suggest shared biological activity. So the process involves selecting a set of chemical features for each drug and then constructing networks based on the shared chemical features. So for example, if we have this particular ring feature we have this many hydrogen bond acceptors or these many hydrogen bond donors. So these are all the shared chemical features. So this is exemplified by the statistics based chem informatics approach. Okay, of course these are some drawbacks. The database, sometimes the errors in chemical structures, it leads to a wrong prediction, and the physiological effects, because always we go for like unmetabolized drug, unionized form of the drug. We always consider the chemical features or chemical scaffold of that particular, if metabolism happens. Okay, so that things and all, it won't be completely told by these softwares. Okay, and this is very interesting, based on adverse effect signatures. Okay, what is the hypothesis? The hypothesis is that two drugs that cause the same adverse effects may be acting on a shared target or protein or on the same pathway. If the adverse effect is same, it is understood that the effect is on the same target or the same protein which is on the same pathway. It is also possible that the adverse effect phenotype of a particular drug may resemble that of a disease. This would suggest shared pathways and physiology by both the drug and the disease. Okay, so these are all some tools, especially the text mining, unnatural language processing, unified medical language system ontology. So these tools will give you the adverse effect. Okay, and molecular docking, computational molecular. So the first signature is over. It may be transcriptomic, chemical, or it may be the adverse areas. Then coming to the computational molecular docking. So it is a structure-based computational strategy to predict binding site complementarity between the ligand and the target. If there is prior knowledge of a receptor target involved in a disease, then multiple drugs could be interrogated against the particular target. Okay, this is conventional docking, one target and multiple ligands. So this is a conventional drug research. What this drug repurposing, what we will do here, inverse docking. That means we take one particular ligand, it is acting on like how many targets it will be. So you can screen a ligand against various targets. Okay, so you can to identify novel interactions that can be taken forward for repurposing. Okay, this is very interesting. So for example, mebendazole, it's a well-known anti-anthelmintic drug. Now it is used like VEG of the vascular endothelial growth factor 2 inhibitor. It is a mediator of angiogenesis used in the various cancer therapy. And the next one is genome-wide association studies, that is GWAS. So this GWAS aims to identify genetic variants associated with common diseases and thereby provide insights into the biology of the particular disease. Okay, so which variants, like where the mutation occurs, what type of mutation has been involved in the particular target. So these all it will be provided by this genome-wide association studies. Okay, so the data obtained may also help identify novel targets, some of which could be shared between diseases treated by drugs and diseases phenotype studies by GWAS, and thereby lead to repositioning of drugs. Okay, so the very good example is denosumab. It is used for the treatment of postmenopausal women with high risk fracture of osteoporosis. Okay, now it is against TNF alpha, that is tumor necrosis factor superfamily member TNFS 11, which is also called as rankle. So here what variant they have identified? They have identified the given RS2062305. It is used for Crohn's disease. And this is very interesting. The next one is network mapping or pathway mapping. So the pathway based or network based approaches have been widely used to identify drugs or drug targets that may have potential in repurposing. So here you can construct the protein drug interactions or protein interactions, okay, based on gene expression patterns, disease pathology, protein interactions or VAS data. So all these things to identify the identification of repurposing candidates. Some of the signature matching studies discussed earlier also make use of this network analysis. So one such example is Cranulocast, a leukotriene receptor 1 antagonist, which is used in asthma. Another one is amrinone, prostaglandin diestrase inhibitor. And the fifth one is by using the retrospective clinical analysis, use of EHR, electronic health records. So the retrospective clinical data can be obtained from various sources, including EHRs or post-marketing surveillance data and clinical trial data. So this EHR contains an enormous amount of data on patient outcomes, both structured and unstructured. So the diagnostic and pathophysiological data, including the results of laboratory tests as well as drug prescribing data, are more structured, as well as it has some unstructured data also. So from this you can do some statistical analysis and data analysis, then you can find out some drugs. Okay, so the one such drug is terbutaline. It's a well-known anti-asthmatic drug which is used for the treatment of ALS, that is amyotrophic lateral sclerosis. So with this we are concluding the various computational techniques. Okay, where exclusively you use various statistical tools as well as the online and offline computational tools. And real techniques, that is real experimental techniques, the binding assays to identify target interactions. So proteomic techniques such as affinity chromatography and mass spectrometry have been used as approaches to identify binding partners for an increasing number of drugs. Okay, then cellular thermostability assay, SETSA technique, like one such example is imatinib, which is used for gastrointestinal stromal tumors. And the second one is phenotypes screening, okay, high throughput screening, and various in vitro techniques and in vivo techniques. So these are all used for the disease relevant effects in model systems without prior knowledge of the targets affected. Okay, so these are the different drug repurposing approaches. Okay, so here what is my view is none of these methods alone will be sufficiently able to disclose the complex interplay between drugs, targets and disease. You have to integrate 2 or 3 computational tools. Okay, one such case study only I am going to explain to you. Of course, all the computational studies have to be validated with real-time experiments and experimental techniques. Okay, so the possibility of using one or more computational approaches to navigate through the wealth of available information and hopefully find clues solid enough to justify a repurposing worth of experimental investigation. So you have to generate the hypothesis, then you can prove the hypothesis by the in silico techniques, 1 or 2 computational tools. Then that tools have to be proven and validated with experimental techniques. So these are the combination. So the choice, how we will select, okay, whether it is say a signature, whether it is a computational dock, or whether it is docking, whether it is a network analysis. So these are all needs. It depends on like the type of the work, the aim of the work, and what is your expected outcome. Okay, so the choice of the most appropriate method or methods will basically nature of the problem to solve and on the time, quality and quantity of information available on that problem. Okay, that's what I told. First we have to identify the problem statement, we have to identify, and we have to generate our own hypothesis. Okay, and obviously we have to depend on the publicly and some proprietary databases for proving our hypothesis. These are all some databases. One such is drug repurposed database. It will give you the drug original indication, what it may be for the new indication, and these are all various databases. If you want chemical structures, you can go for PubChem. You can suppose if you want the drug, you can go for Drug Bank. Okay, you can go for Drug Map Central. Okay, I want the target 3D, three-dimensional structures, of course protein data bank. Then drug target information, I can go for therapeutic target databases. These are all some likely publicly or proprietary available databases. Of course, the drug repurposing has got its own limitations. Okay, the first limitation is, though so many pathways and so many mechanisms are explored, there are some diseases which the mechanisms are not explored. The pathways are not known. Okay, so the pathways and mechanisms of many complex diseases remain incomplete, and efforts for drug repurposing often require analyzing complete data. And the second one is, I told already, we are completely believing that on the databases. Okay, sometimes we won't get sufficient information from the publicly available database. Suppose for example I wanted to do a particular protein, the protein structure is not available on the database. Again I have to do the homology modeling, so there may be some difficulties. Okay, and again, there is no systematic mechanism to obtain fundings to support drug repurposing. Of course, we have sometimes the call for, from in India, I think some 2 years before, year before, there is a call for drug repurposing, but still we have some lacuna compared to the traditional drug discovery project. Okay, of course, we have the patent issues, okay, especially the market exclusivity issues. We have to go for new method of use patent only. Okay, because already the drug has been patented for its own use. Okay, one use. Now again we have to go for a second use. We have some patent issues. Of course, we have some regulatory challenges. Okay, so with this I'll be concluding my first part. So till now, is there any doubts, or shall I continue with my case study? Hello dear participants, any one of you can give me the signal.
That's right, Office. Continue.
Yeah. So my case study is about an in silico drug repositioning approach of identification of drug candidates for breast cancer therapy. It is the work carried out by one of my scholar, now he's a doctor, Dr. Naharaj. Actually, we have utilized 3 techniques over here. One is the networking pathway mapping. The second one is structure-based drug design. We have used molecular docking, dynamics. Then the same thing has been validated by in vitro techniques. Okay, these are the 3 techniques we have used. So what is our hypothesis? Actually, the first and foremost thing in the drug repurposing, the goal of this research work is to identify the drugs for the suitable targets of breast cancer pathways through in silico drug repurposing approach. Okay, so the collection of information, the right side, we collected the information, okay, of the drugs, targets, proteins, genes and pathways. Then followed by that, the construction of network, drug similarity network, target similarity network, heterogeneous network. Then random walk, prediction of drug target interaction. Then identifying the repurposing drug candidate. If it is on patent, we can use as it is and we can go for reformulation. If the patent, the patent, that is the expiry, if off patent, again use as it is, and commercially driven development, IP production, rebranding, etc. And finally, new market authorization, label extension of label use. So based on the hypothesis, we have the aim. What is our aim? This research aims to identify